This is a production of Cornell University.
Thank you, Gillian, and thank you for the invitation. This is, I would say, this is probably my first full seminar that I've done this way. So while I'm disappointed that I won't hear all the laughter from my hilarious jokes, I have a good imagination, so I know that you guys are all infotained. So it is a great pleasure, and as I was preparing for this, I realized, and I emailed Gillian yesterday, we've known each other now for 2 decades, which is, our friendship is almost able to drink legally. I'm really excited to be talking about genes and phenotypes. So the outline for the talk is I will first talk about EMSL, and how it works as a user facility, what we can do in collaboration with you, and then I'll go into some of my own research, all kind of talking about genes to phenotypes and phenotypes to genes. And without further ado, I'll just start. This is a picture of EMSL. It is, I think it's the size of about 3 Costco's, is what I've heard. It's a massive lab in the middle of nowhere in Washington State, about 250 miles east of Seattle, along the Columbia River, in a smallish community where the Department of Energy or its precursors made plutonium for nuclear weapons. Now we're involved in cleanup. And EMSL is a part of the Pacific Northwest National Lab. We're a DOE lab that focuses on all of DOE's missions, including basic research. And so EMSL is this user facility, and it was founded in 1996. So we're getting close to our 25th anniversary. We're funded out of the Office of Biological and Environmental Research, part of DOE, and we address science problems that are relevant to energy and environment that can use analytical chemistry, math, computational sciences, some combination of that to accelerate research and solve problems. And our budget's about $44 million a year. We support over 700 users per year. And I'll just say that we basically I run peer review processes for many or most of the projects that come through. So it's a competitive process. It's open to anyone, and we really focus on DOE mission science. And in this case, it would be biotechnology, biofuels, plant genomics, so things that are probably in all of your wheelhouse. So I'll talk a little bit, just a brief background. Functional and systems biology is the area that I lead. There's a kind of parallel area in environmental research that's also makes use of the same kinds of tools. My area is really interested in omics and microscopy that support understanding, say, carbon cycling via microbes and plants. At the molecular level, also biotechnology for biofuels. So I like to say anything that you need to do after you get a genome for your organism, we can help you with. And we're organized into integrated research platforms, and I don't want to go into too much depth about how we're organized, but suffice it to say we have capabilities that span all kinds of imaging, from light to electron microscopy. We have a brand new cryo-EM, so we're getting into protein structure. In cellular dynamics, we're interested in what it sounds like, how cells are dynamic. We have technologies to look at cells, do some level of omics with single cell resolution. I'll talk a little bit about one of those technology. And then functional omics is really your proteomics, metabolomics, glycomics, transcriptomics, pretty much any omic. There's a lot of them out there. And our focus there is we have NMR and mass spectrometry, as well as some sequencers. And so we do all kinds of different permutations on this. I can only touch on a few things, but I'm happy to elaborate. If you want to email, I can send information, and you can also go to our website. So we just went through a road mapping exercise, and in biology, we are developing capabilities, technologies, expertise that will lead with the big audacious goal of what we call the digital phenome platform. But really, what this is a research platform that would allow prediction of phenotype based on any number of experiments we could do to get at protein function. For example, we have computational chemistry capabilities that can really delve into enzyme active site chemistry. We want to visualize metabolic pathways. So we want to not only understand what's doing what proteins and other small molecules are doing what they're doing, but where are they in the cell? How are they interacting with their environment? Visual proteomics is a really exciting platform we're developing. Think of it as a space-filling model of a cell where we're combining protein structure information with location in the cell with protein complexes to really kind of pack that cell and conceptually pack that cell and understand what the phenotype based on how that cell is packed with proteins and metabolites. So we use a number of different capabilities to answer questions. In our own research, we can develop genetics, but we have, as I mentioned, ultrastructure, we do microfluidics. We're interested in incorporating modeling with our experiments. This is, let's see if that rotates now. This is super resolution light microscopy of Yarrowia lipolytica, a yeast that accumulates lipid. Those are 2 lipid droplets in there, and then a calcifluor stain for the cell wall. And you can see some nice bud scars there. And then we worked with users to combine all this data into kind of an integrated picture. And we're developing tools along all these platforms. See, here we go. So this is just a quick example of a user project. This is one where we were looking at Neurospora crassa, and we did kind of relative quantitation of the full proteome of Neurospora crassa across a circadian time course. So every 2 hours for, I believe, 48 hours in triplicate and with a mutant. And working with Jennifer Hurley and Jay Dunlap. Jay's at Dartmouth and Jennifer's at Rensselaer Polytechnic Institute. And what we found essentially was nothing too surprising. We were able to identify a number of circadian proteins whose level changes across time. And the I would say not surprising result, but one that was important, was that circadian transcripts don't necessarily lead to circadian levels of proteins, and vice versa. If a protein has a circadian abundance, so it goes up and down with a circadian rhythm, it doesn't necessarily mean that there is a transcript that does that as well. So what it really points to is post-translational regulation of proteins over time, and really the complexity that's involved. And so we're working, I mean, obviously this was a massive project. We've been working with Jennifer and Jay over the last few years, and we've looked at metabolites over circadian time, and these are still kind of in analysis, unpublished results, but also the circadian phosphoproteome. So I put this out there as an example of some of the types of research and the scale of research that we can do. Some of our long-term goals are to automate some of these platform so that we can increase our throughput. So we also can do modeling and are able to work with users. Some users want to collaborate with us and get additional grants, and so that's what happened here. We're working with this team on an NIH DOE modeling grant. This is one of our really cool instruments. It's one of 2 in the world. It's a 21 Tesla FTICR mass spectrometer. It has amazing resolution such that the mass accuracy is able to is so high that we can identify compounds, molecular formulas with really high confidence. The 21 Tesla refers to the magnet, and this was a custom magnet that was built by Agilent, and then we also design our instruments. So this mass spectrometer was actually designed and built in our lab. We have fabrication capabilities, but really it's cool to look at. But what can it do? So this is a real brief experiment. We had a mutant of Yarrowia lipolytica, and I'll be talking more about Yarrowia later. But Yarrowia is a yeast in the Saccharomycetinia that when it's grown on low levels of nitrogen, high levels of carbon, or high carbon to nitrogen ratio, it starts to accumulate lipids, which can be converted easily to biodiesel. So it's of interest to DOE. And we were doing mutant screens and found that we could make what normally is unpigmented Yarrowia, we could make it red. And so we worked with one of the developers of this and found that we're basically able to isolate the red pigment, shoot it into the mass spec, and came up with a formula basically came up with a mass that was consistent with this red pigment being protoporphyrin, which is the last step of heme biosynthesis before iron is added by ferrochelatase. And so we looked at the isotopic fine structure, so you can not only see the high abundant isotopes, but also your 13C and other all the different stable isotopes that are naturally, you can see them in their natural abundance. This was a sigma protoporphyrin that we were able to buy as a standard, and you can see that the red compound from Yarrowia has the exact same mass spec. In fact, it's a little bit purer. We were able to isolate a more pure isolation of this for our experiments. So I'm going to show one last kind of capability and then go into more of my own fungal research, but we're working on capabilities to look at transcripts in cells using fluorescence in situ hybridization. And so we're able to use different combinations, use different combinations of fluorophores to look at, right now, up to 10 genes in a single pass using fluorescence in situ hybridization. And this is what it would look like. This is actually, I believe this is a mammalian cell where they're looking at genes of interest in insulin production. But basically, we can look at 10 using kind of a fluorescent barcode. You can look at the transcript levels within single cells of up to 10 genes, and we're working to multiplex this so that we can look at 50, the expression level of 50 genes at one time. And so imagine now that you're able to look across a microscope slide, you can start to look with single cell resolution at transcript levels and start to look at how do transcript levels vary across a population of genetically identical cells. So one last advertisement, COVID's obviously on everybody's mind, and in the last stimulus, we got $3 million for capital equipment and used it to purchase some mass spectrometers and some capability for our cryo-electron microscope. And we also got funds that allow us to actually do COVID proposals. So if any of you have, we still have some of these operational funds for small projects in the COVID arena. Contact me if you're interested in proposing. We have a kind of a rapid review cycle, and again are looking for have the mandate from DOE to actually do COVID research using some of our equipment. And then finally, we do a lot of outreach, and we'll be having a summer school for graduate students and postdocs, and then with seminars mixed in for everybody to look at taking multi-omic data, modeling it, and visualizing it. And so stay tuned for that. We had a summer school last year on microbial modeling. It was free. A lot of sessions were free. We have some small amount of competition for like in-depth classes. It's a week long. It's the last one was really quite valuable and exciting. So we're hoping for this to be the same. And the idea again is to kind of get an in-depth view of tools for multi-omic modeling of biochemical pathways. So I'm going to go into more of my research, and I wanted to start with an acknowledgement. These are really, a lot of these are very large projects, and it wouldn't be possible without a team. So at EMSL and PNNL, I'll be highlighting the work of two now staff scientists, former postdocs that stayed on at PNNL, Aaron Bredaway and Kyle Pomranning. The Aspergillus genome work was co-led with Mikael Anderson of DTU, who's now at Novozymes. All the sequencing was done at JGI, and Igor Grigoriev is the head of the fungal genomics program. Ronald DeVries at the Westerdijk Institute has done a lot of the growth profiling. And then I'm also a part of the Joint Bioenergy Institute. So I have an affiliate position at Lawrence Berkeley Lab, which leads jbei, and John Magnuson, a PNNL colleague, actually is the director of the fungal biotechnology team. I just get to do science, which is kind of nice, no administration there. But we have a small team there working on fungi. So you'll see an amalgam of all these, this team of teams to really do fungal biology and connect genes and phenotypes. So I had no idea that when I picked this title, I guess I was prescient that flies would be trending due to the vice presidential debate. So as Gillian mentioned, I did a postdoc looking at fly genetics before I saw the light and went to fungi. And so I threw it into the title, not knowing I would be so trendy. But I'll tell you where I got my start in connecting genes and phenotypes. And just a little graphic to show I'm interested in that. It's been an interest of mine since I started school in biology of connecting genes and phenotypes. So I did my undergraduate work at Oregon State, and that was really where I thought, oh man, these fruit flies are really cool, great genetic system. Little did I know the kind of ins and outs of fly husbandry until my postdoc years. But in any case, I did a postdoc, and before there was a genome for Drosophila that was publicly available, so we're talking, I guess, late 90s here, I did a screen where I looked for suppressors of a dominant wing blister phenotype. And I think I have, I was probably one of the last people to find their gene before genomes came out. There was obviously beautiful mapping in Drosophila. Basically, we found that a signaling pathway was impaired and it caused blisters. And so in this mutant phenotype, and so then when we killed that signaling pathway, the flies were, I wouldn't say killed it. When we made hypomorphic mutants in the signaling pathway, we were able to suppress the blistering. And it was actually turned out to be a nuclear import factor that's able to bring MAP kinase into the nucleus. But I want to dwell on Drosophila. Let's go to fungi. And I tend to have as Gillian mentioned, a fungal-centric view of at least my professional life. Fungi are, as I'm sure many of you will agree, are really amazing organisms. And I started in fungi kind of on the faux side, thinking about plant disease, working with Gillian on Cochleobolus. But then when I went to PNNL, my focus changed towards biotechnology and using fungi for enzyme production or small molecule production. And from there, it's been a lot of fun to sort of, like I said, connect genes and phenotypes. So, some of these fungal phenotypes are secretion of enzymes, secretion of useful small molecules. So there really is a nice connection to biotechnology and, in a lot of ways, biofuel production and biochemical production, which are central in DOE biology research mission. So I'm just going to launch right into it. One of the projects that I started a few years ago, actually several years ago now, was through my Joint Bioenergy Institute connection, and that was to sequence every single species, one example of every single species of Aspergillus, so that we could really look at the whole genus of Aspergillus and understand evolution. And so my co-PI for this was Mikael Anderson. We sequenced, right now there's about 220 strains. There's several that are kind of winding their way finally through the pipeline, but most of the sequencing now is complete, and we're starting to look at different sections of Aspergillus. The first section, that we looked at was Aspergillus section Nigri, which are the black Aspergilli. And you can see Aspergillus niger is one of the key species. It produces citric acid as well as a number of enzymes for industry. And we can now look and develop phylogeny based on the whole genome. You can see there's, in this column here, is the number of predicted proteins. It varies even within this small section of Aspergillus. There's quite a bit of variability, as well as variability in the genome quality. One of the phenotypes that's interesting about Aspergillus is that Aspergillus niger produces fumonisant, and we discovered this when we were sequencing the Aspergillus niger genome probably a decade ago. We saw this cluster that looked like the Fusarium fumonisant cluster, and it brought back memories of my Cochleobolus years, because we'd found a fumonisant cluster in Cochleobolus. So when I published this, it's not within our mission to really look at secondary metabolites, but there was a group at DTU, Danish Technical University, that led by Jens Friesvad, that became very interested in the fact that Aspergillus niger, which is this industrial organism and considered generally regarded as safe for food production, etc., Aspergillus niger might make fumonisant. And so they went and actually found that niger makes fumonisant, and then looked at a whole bunch of different foods where Aspergillus niger has been found: dried fruit, coffee, chocolate, etc., looking to make sure that fumonisant was not making its way into the food supply in that way. So there's a whole bunch of papers on that. When we sequenced all of section Nigri, we could actually look and see what percentage of the fungi in this section have the ability to make fumonisant. What we found was that there were a few closely related species to Aspergillus Niger, as well as one that's closely related to Aspergillus carbonarius that have what I would call the full fumonisin cluster, and then several that have just the polyketide synthase associated with fumonisin. And then one other thing we noticed was a second polyketide synthase for which we don't know the metabolite. Always seems to cluster near the fumonisin PKS. So again, connecting genes and phenotypes, we can look at the secondary metabolite cluster or secondary metabolites associated with each section of Aspergillus, and then try to draw correlations with which secondary metabolite cluster might make that particular metabolite. So obviously sequencing genomes kind of gives you the potential, but then you have to dig in and do a lot of the hardcore biochemistry, which is continuing with the group at DTU. So we've also just recently published in Nature Communications section Flavi, which has some wonderful Aspergilli, Aspergillus oryzae, Sojay, so some really beneficial Aspergillus species that are used in food production in Asia, so the koji fungi, but also some infamous plant pathogens, Aspergillus flavus. So in this clade or section actually of Aspergillus, we have aflatoxin production. We've again looked at the secondary metabolites and assessed whether which metabolites are made by which particular species, and then started again to look at the number of potential backbones associated with these metabolites. I think one thing that is clear is that the genetic potential for these organisms when it comes to secondary metabolite production is vast, and we really don't have a great grasp of all the metabolites, and those connections to the secondary metabolite clusters still need to be made. What we've also noted, and this is kind of a busy slide, but I can sum it up to say that as many of us know, fungi have an incredible capacity, enzymatic capacity to deconstruct plant biomass. And so when we grow these aspergilli, on different carbon sources, their growth ability is quite, doesn't necessarily correlate with the carbohydrate active enzymes or Kzymes that are in the genome, indicating a really critical role for regulation of these genes in the lifestyle of this group of fungi. So while these are very closely related, they grow differently on different substrates, despite having pretty big Kzyme potential, and that's again getting to that phenotype of whether something, how it grows, and relating it to genes. It's much more complex than just being able to sequence the genome and look at the catalog of Kzymes. So I think this is, yeah. So then finally, some unpublished data from section Usti. Again, the point illustrating that we can kind of make phylogenetic trees based on the genes, and then on the genes in each genome, and then we can see how closely related they are, and that obviously more closely related Aspergilli have very similar secondary metabolite gene clusters. Again, there is still tremendous diversity even within one section of Aspergilli. So summary for this section is to say that the whole genus genome project has kind of given us a view of the genetic and chemical diversity with single species resolution, tremendous chemical diversity, and regulation is a key component that is a differentiator between species that may have similar catalogs of genes. There are two other whole genus projects that are starting up, one in Penicillium, which is about 500 different species, and then another one led by Randy Burka from formerly of Novozymes, now at ADM, on trichoderma. So stay tuned for those. Again, I'm going to talk about connecting genotypes and phenotypes, and I'm going to talk a little bit about Yarrowia lipolytica. This wonderful oleaginous yeast. It's actually a dimorphic yeast. It grows in yeast form and then hyphal form, depending on conditions. It's got reasonably good molecular genetics, not as good as Saccharomyces, but still pretty good. And there's metabolic models available, and then several strains have been sequenced. So this is a movie of Yarrowia growing. It's a time lapse, and what we've done with this particular strain is that we've labeled the GF with GFP, although it's showing up red here, lipid droplet protein, and you can see lipid accumulation over time in this organism. If you look at this guy over here, you can see it's kind of pretty cool. You can see here's the lipid droplet, and then as the daughter cell buds, you can see the lipid droplet divide as well and move into the daughter cell. This is the kind of gross response to nitrogen limitation, and we started to kind of explore the regulatory pathways involved in not only lipid accumulation, but the morphologies associated with it. So the morphology is really important, and if you talk to anybody at an industry that uses filamentous fungi, especially in their work and to produce enzymes or metabolites, they'll tell you that morphology is absolutely critical in the fermenter. As you might imagine, if a fungus kind of grows in these long hyphal masses, it becomes, and you've seen it maybe in shake flasks, it becomes very viscous, and it's not really an optimal bioprocess. And so Yarrowia can make hyphae in bioreactors, and that can kind of really gum up, not to use an unscientific term, the process. And so one of the first things we did in this Yarrowia process or project that when Kyle Pomeranning was a postdoc in the lab was he did a screen for mutants that don't make hyphae. And what he found was you could look for, just at the colony morphology, you'd get these what we call smooth colonies. And so he did, Kyle did a screen of about 500,000 colonies and isolated 65 mutants that were smooth. And then of those mutants, he picked 5 that did not make hyphae under any of our tested environmental conditions. And so obviously, one of the things we want to do is to understand what mutations cause this. This is just a little bit more microscopy looking at the smooth strains. You can see, here's our wild type, and you can see the yeast form in our smooth mutants. And one of the things we did was to sequence the genome of these smooth mutants. And I'm going to kind of sum up a few years of work in the next few slides. Basically, with these smooth mutants, we found that it was not a simple gene hit or mutation in a single gene that led to the phenotype. What it was actually was in a subtelomeric region, we started to make, in 4 out of 5 of our smooth mutants, we started to make deletions of repetitive DNA. And basically, long story short here, you can see that, this is the easiest one to look at, is this bottom column here, that the repeats decrease in all of our smooth mutants. So the repetitive region, the number of reads that contain repeats when we do sequencing decreases in a couple of mutants, or actually all 5, but in 2 in particular, our presumed amount of rDNA in the genome also is decreased. And I should note that these smooth mutants were made in a KU background. So we've gotten rid of non-homologous end-joining DNA repair, and there is some kind of evidence out there that sometimes these mutants can start to accumulate different kinds of deletions in repetitive areas. So we wanted to continue to go after this, and one of the things we did was to then do a suppressor screen using one of our smooth mutants to look for then suppression of this smooth phenotype. So how do we restore hyphal growth? And one of the things we found was that we started to accumulate mutations in histidine kinase and MAP kinase signaling pathway. In particular, NIC1 and CHK1, these are the 2 histidine kinases where we saw mutations. And what that led us to then was kind of a high-level view that in the KU mutants where we've accumulated these deletions in the subtelomeric regions, we're causing some sort of stress that is activating these 2 pathways, the HOG-MAP kinase cascade and then some histidine kinases that essentially then feed into, and this is results I didn't talk about, the transcription factor MSN2 that lead to this change in morphology. So again, what we did was, looking at these mutants, we were able to use high throughput or next gen, it's now current gen, I guess, sequencing technologies to quickly get at the mutations and start to associate genes with our morphology phenotype. So we're continuing to do a lot of work in this is kind of the future directions of the Yarrowia work. We're doing a lot of more synthetic biology where we try to work through our design, build, test, learn cycle to develop Yarrowia to make other kinds of molecules like organic acids or terpenoids that may have use as biofuels or bioproducts. So the last part I'm gonna talk about is about enzyme secretion. And so I always like to tell people, especially if I go into elementary schools, to talk about fungi and the wonders of fungal research, that we all live in the fungal digestive tract. So fungi have this great quality of essentially digesting the environment around them and then absorbing those building block nutrients versus like humans ingesting food and digesting it internally. So we all live in the digestive tract of fungi, and that's one of the great qualities that led us to use fungi as a protein production platform. So at the Joint Bioenergy Institute, the goal was to develop some fungi that could be used to produce thermophilic bacteria enzymes that could then be used in biomass deconstruction in the background of ionic liquids. So ionic liquids are used at jbei to solubilize biomass, which is then converted into sugars and fermented into fuels and chemicals. So we needed a protein production platform. We took a couple of different thermophilic bacterial beta-glucosidases and put them in, expressed them in Aspergillus niger, and then developed a screen for secretion of this heterologously expressed protein. So I'm going to zip through this section. Basically, what we found was when we did the mutant screen is that we were able to, even in a background of fungal biomass degrading enzyme activity, we could increase the percentage of ionic liquid in our solution, and only the bacterial enzyme really could survive that kind of condition. Fungal enzymes were not good and even as low as 5% ionic liquid. So they in our assay for beta glucosidases, the fungal enzymes would die. So we could do a mutant screen, use a plate assay, which I show here, to look for increased secretion of our heterologously expressed enzyme in a background of fungal enzymes, because as I mentioned, the fungal enzymes don't survive ionic liquid. Basically, we found two types of mutants, and those mutants were either the endogenous enzyme increased, so this is in our beta glucosidase assay, or we saw an increase in the heterologously expressed enzyme. So on the black bar is non-ionic liquid conditions, and then under high ionic liquids, so this is higher fungal enzyme, this is higher heterologously expressed enzyme, and then this is when both sets of enzymes, both the fungal enzymes and our heterologously expressed enzyme, are increased in expression. And I'm gonna kind of cut to the chase here. We sequenced a whole bunch of mutants, and we found that several strains that were high for only the heterologously expressed enzyme had a mutation in a monosaccharide transporter MSTC. We grew these under high maltose in the media, and so under this particular condition, we could then look at this heterologous expression. I mentioned the maltose because this is a predicted sugar transporter, and essentially what we found was that our heterologous, what we ended up screening for, you kind of get what you screen for, was that our construct for heterologous expression was under the glucoamylase promoter, and so we were essentially screening for not necessarily an increase in the secretion pathway, but regulatory mutants for glucoamylase induction. And so that'll figure into, I'm going to skip this and go to kind of the last little part of the message here, which is that we not only did our own screen, but then we could go to strain collections and look at existing protein production strain, as well as some Neurospora crassa classical mutants, and resequence all of them to kind of get a picture of what genes are involved in enzyme secretion in filamentous fungi. And so these are just some of the lineages that we sequenced. So here's Trichoderma reesii, famous for, it's probably the parent strain for a number of enzyme companies and their biomass enzyme cocktails. The reference is OSQM6A, and then there's this well-known lineage, most of which is deposited in ATCC of several high-producing strains. These are some Aspergillus niger strains that produce high levels of glucoamylase. And I'm going to get to the punchline here. And these are some of the genes we found. There's known regulators of, we found a bunch of basically known genes, CRE1, ACE1, CRE-A, amylase, AMI-R, and the bottom line, and then we also found other sugar transporters that had been mutated. The bottom line is that the way to make a hypersecreting fungus is to, one, go after the transporters and sugar sensing machinery, and then to mutate the transcriptional regulators. And then there's also some good data that you can impact the secretion pathway. So what you can do is you can stack these kinds of mutations to get what essentially are fungi that are secreting industrial levels of enzyme. So with that, I'm going to end. Hopefully I've left enough time for questions.
Thank you, Scott. My head is spinning.
I was worried about packing. I actually got rid of some information, some slides at the last minute, so probably a good thing.
Before I forget, everyone, all graduate students and postdocs are invited to meet with Scott. We used to call this a lunch. No longer a lunch, but you're welcome to eat. Starting at 1:45, I believe, and there's a separate link that went out. Let me look at the chat. From Claire Castile, maybe I missed this. Do the hyphae mutants have any other changes in phenotype? Mutations in stress signaling seem important.
So yes, the colony, obviously the colony, the colony morphology is changed, and you'd think my throat would be used to long Zoom meetings. I'm a little bit scratchy now. There's also, these are also pathways involved in, so stress response to osmolarity. So we haven't really put them through their paces to see all the, I think the phenotypes are pleiotropic. Like I said, I can tell you that not all of the smooth mutants are super healthy. So I'll just leave it at, yes, there are pleiotropic phenotypes. I didn't talk about them, especially in the osmolarity signaling and stress response.
So I was going to ask a similar question regarding your last slide. You make industrial levels of enzyme, but are you alive?
No, these are pretty unhappy strains. You know, the beauty of trying to figure out these genes is, so the Trichoderma ricei, the kind of strain that started it all, is actually female sterile, and so the multiple rounds of mutagenesis just piled up mutations, and so it's a pretty unhappy strain. I mean, I guess they can always get worse. So that, I think when you start to pile on these more directed mutations, you still make them sick, but not as sick. So that's kind of the goal, is can we get rid of the collateral damage and try to make them healthier? But no, they're not happy.
Second question in the chat. In the Yarrowia project, histidine kinase was found to be related to smooth mutants. What is the biological function of the histidine kinase in the organism after phosphorylating histidine?
So I know that it's kind of in that upstream stress signaling pathway. Some of we haven't done a lot of finer phenotypic mapping, so I can't really answer that question because I don't know. Just generally, histidine kinases are involved in kind of signaling these important, I think, osmosensing. I think there's some of histidine kinases are also involved in they get mutated and become resistant to fungicides. I'm trying to remember the class. We started to look a little bit at this in some of our other work. But suffice it to say, these cells are pretty sick. I have kind of an unsubstantiated theory that when we made the KU mutants and then screened these KU mutants for there wasn't really a heavy mutagenesis. So I think we picked up mutation, or we picked up strains where the DNA damage, because it was in a KU background, was so great it caused it started to mess up the regulatory pathways in ways that go beyond our genetic screen. So I wish I had the funding to follow up on this. That was a long-winded answer to I don't know. Cool stuff.
That's okay. Another question. Very cool work. Thank you for the presentation.
Thank you.
General question relating to the first half of your presentation. Has EMSL or have you done work with syncytial cell imaging or omics? I guess it would technically fall under single cell omics, but they often hold bacterial cells inside the cytoplasm. So I'm curious if anyone is trying to tackle this.
So trying to make sure I understand the question. Basically, are we doing...
Feel free to unmute Marina and...
So syncytial cell imaging. I'm a little, so looking at, like, for example, bacteria as symbionts inside of other cells, or?
Hi, thank you. Yeah, I was mostly curious because you mentioned the work on sorry, what was the term? Your flyfish work?
Oh, yeah.
Yeah, and I was just curious if anyone was looking at syncytial cells where they have like the combined nuclei in a large cytoplasm.
So, yes.
Okay.
So we've been working with Jonathan Schilling, who looks at white rot fungi, degrading wood. So yeah, I could go through different user projects, but the answer is yes. We are looking at shared cytoplasm and with multiple nuclei. We're also just to advertise, we've done some studies where we've looked at endophytic bacteria in poplar. So there's a lot of so the answer is yes. We work with users to customize kind of extractions and fixations and so forth, because no one microbe is the same, or plant.
Okay, thank you.
Yeah, follow up with me if you want to.
Really cool.
Thanks. And it's accessible to everybody, so follow up with me.
This has been a production of Cornell University, on the web at cornell.edu.